Hello, welcome to DM Neurology Made Easy. Today is the 7th episode of Fine Neurophilia Quick Revision series. Here we will discuss some core concepts of dementia in Dementia Part 2 video.
This class is basically to solve this concept. This question is the basic one question which I wanted to clear. The concept behind this question was the one I intended to clear this class on. As you know, amyloid precursor protein, we have heard that it is in chromosome 21. It is associated with Alzheimer's disease. So what do you think? Amyloid precursor protein in Alzheimer's disease is cleaved by all except. This concept has been there in Harrison as well as Bradley. I want you to know about it actually. So in Harrison and Bradley, about Alzheimer's disease, you know, there is actually two causes. That is why Alzheimer's disease occurs is because of these two things, amyloid plaques and neurofibrillary tangles. I have tried to simplify it for you. These amyloid plaques, as we can see here in neurons, there is normal neurons, there is nothing you can see. But in Alzheimer's disease, you can see these amyloid plaques, which are actually extracellular. These are neurons, so extraneuronal or extracellular. And in neurofibrillary tangles are within the neurons. That is, NFTs are within the neurons. So intraneuronal or intracellular. So these all can come as sentence questions, true or false kind of questions. So amyloid plaques are extracellular, neurofibrillary triangles are intracellular. So why this happens? Amyloid precursor protein. And as you have seen, it is present, it is originating from the chromosome 21. What is this function of this amyloid precursor protein? It is actually a normal poor protein which is present within the neuronal membrane. It has a function within the membrane. The function of this protein within the membrane is to produce synapses. Initially, in the first question, we had seen that all the neurons has to produce synapses, has to have a neuronal migration, because it has to develop. For a child to develop into an adult, it has to have neuronal migration. Also, it has to help in transport of ion. All this, example, ion transport, that is ferric ion transport, all this is helped by this APP, amyloid precursor protein. So this is present on the neuronal membrane. But what happens is that once this APP is about to die, it has to be normalized and disposed of. So normal cell has got certain ways of cleaving it off and disposing of without any much of a trouble to the body. So this is actually regarding the function. So synaptogenesis, neuronal migration, cell adhesion, and also ion transport. So any of this is affected, then the cells become damaged. So ion, if gets accumulated, it causes neurotoxicity, etc., etc. So what happens? That was regarding amyloid precursor protein. So when it becomes old, it has to be degraded. So now our body has normal methods to degrade it off. There are two pathways. One is the non-amyloidogenic pathway. The other is the amyloidogenic pathway. In non-amyloidogenic pathway, two scissors cut these APP into two parts, alpha and gamma, which are, I don't want you to remember the names and all, but they equally fragments and easily gets disposed of. No problem regarding that. In amyloidogenic pathway also, usually there is no much of a problem, but usually it is acted on by beta and gamma. So here what happens is because of this amyloidogenic pathway, there is something called amyloid beta monomer, what we call it as A beta, which we usually hear A beta. A beta is produced in a normal human being without any problem. Usually this amyloid beta will be cleaved by the microglia, the other astrocytes, and some enzymes, etc. will cleave these cells, and there will be no much of a problem. But in case of some mutations or some disease and all, what happens is that this A beta increases in number, and they become beta oligomers. They clump together and become beta oligomers. And that is how these senile plaques or amyloid plaques are formed. And amyloid plaques, once formed, as we have seen, they are present in between the cells, and they block the neurotransmitters. So neurotransmitters are needed for transmission within the cell, in between the cell, and they block the neurotransmitter and hence block all the synaptogenesis, all the synaptic transmission, etc. So that is the problem with the amyloid plaques within the cell. So that is regarding amyloid plaques or senile plaques. Now we told about one more thing, that is actually neurofibrillary tangles. So what we have seen is that actually inside the cell, you have seen that there is a soma and there is an axon. We have seen a soma and axon. So in the cell body, every material has to be brought into the end of the dendrite, and also some things have to be brought from the end to the soma. So this is acting or occurring through a pipes called microtubules. And these microtubules are strengthened with the help of tau. This actually, you can think, analogy can be given to these pipes strengthened with tau. These tau can be thought of as ropes. So tau has got this phosphokinases are there in tau, which binds these areas together. So what happens is that due to disease, this kinases become hyperphosphorylated. So you think that these ropes become broken. What happens to the when ropes become broken? Actually, these microfilaments or tubes become broken, and these taus become the ropes become hyper accumulated together. That is hyperphosphorylated tau becomes accumulated. That is neurofibrillary tangles are formed. NFT, neurofibrillary tangles are formed, and these tubes, microfilaments becomes disintegrated. So there is no transport. There is actually no anterograde or retrograde flow within the cell. So the cell usually dies. The neuron dies. So this is what happens within the neurons. So these two things, that is senile plaques or amyloid plaques, neurofibrillary tangles, occurs within the Alzheimer's Now, if you see this picture in Bradley, I think it will be more easy. In non-amyloidogenic pathway, you saw alpha and gamma. It cleaves to produce non-toxic particles. In amyloidogenic pathway, also beta and gamma, it also produces non-toxic particles. But sometimes this A beta gets accumulated to produce plaques. It's very simple as such. There is no confusion. So this actually is a simple diagram. And Alzheimer's, one more thing is that there is enzyme deficiency. This enzyme is deficient. That's all. That's why we give donepezil to supplement for this enzyme, because this neuron is damaged. So dysfunctional cholinergic neurons. So we try to supplement it with the help of donepezil. So this, if you have got this pathophysiology right, every question coming from after this, that is any question coming after this pathophysiology, you will be able to answer. Biomarkers of neuronal injury in Alzheimer's disease include all except, is it increased tau, increased CSF beta for A beta 42, hippocampal atrophy, abnormality in the FDG PET metabolism. So actually CSF beta, A beta 42 is increasing within the brain, but it is not seeping into the CSF because it's getting clumped and increasing in number. And what we actually see in Alzheimer's disease is actually a ratio of A beta 42 versus A beta 40. And as you saw, that CSF tau is actually our clumped drops we had seen in that. And hippocampal atrophy is actually the net result of all the cells dying. And also, because the cells are dying in the MRI or the FDG PET, we get the hypometabolism. So we think that there will be CSF A beta 42 increase because A beta 42 increase during the pathophysiology or the damage of in Alzheimer's disease, but A beta 42 increases and clumps together and gets stuck within the brain and doesn't seep into the CSF. But the others, A beta 40, A beta 37, A beta 38, etc., seep out. But A beta 42 does not. So if you see, ideal thing is to see the ratio of A beta 42 to A beta any others. So best is to see for A beta 42 by A beta 40. Hope you got the concept. And in Alzheimer's disease, routine CSF is normal. So your routine things, blood cells, proteins, etc. are normal. So which abnormality will suggest, this is actually a repeat question which I am asking you, which abnormality will suggest that the patient has got Alzheimer's disease? I told you actually CSF tau won't get reduced. Tau actually comes down through the CSF and it will be increased. So remember that CSF A beta 42 will be reduced because it's clumped and stays within the brain. CSF A beta 40, 37, 38, 80, etc. will increase along with hyperphosphorylated tau. So remember that concept. I just put that question to reinforce that fact. Alzheimer's disease, cognitive changes follow a characteristic pattern. So as we have seen with the pathophysiology, there is atrophy, the neurons get damaged. Where is the maximum neuronal damage? And what is the first thing which we see in Alzheimer's disease? So there is a pattern of cognitive change. So there is a progression of the disease from mild to moderate to severe in case of Alzheimer's disease. We call it by a name of this kind of progression. I'll tell you later what is this kind of progression we call. It's called a staging is there. It's memory followed by language, followed by visuospatial, then executive. Temporal followed by inferior frontal, then parietal occurs, then dorsolateral prefrontal cortex gets affected in Alzheimer's disease. So this is the correct sequence of progression. So if you see, this is mild. First, there is actually short term, followed by language. All we have seen. So this table, this actually I had taken from Harrison. The page cut. This page has been taken from Harrison. And this staging is actually called a Bragg staging of diseases. So Bragg staging of diseases, if anyone can comment in the chat box, you can do that. Is used in which disease actually? Any other disease in which Bragg staging is used? Bragg and Bragg staging. These are the 6 stages are there in Bragg and Bragg staging. That is transentorhinal stages in which the patient will be asymptomatic. Then the limbic stages, then it spreads in the cortical stages where the patient will be having the major cognitive impairment. So which of the following is not a atypical variant of Alzheimer's disease? So we have seen that there is a typical variant of Alzheimer's disease in which the disease spread from the temporal lobe to inferior frontal, then the parietal, and then to the dorsolateral prefrontal, and the occipital lobe gets affected the last. So when there is a change in pattern of such kind of an occurrence, so if actually occipital gets affected first, or if language area gets affected first rather than memory, or what do you call it, frontal area gets affected rather than memory, then we call it as an atypical variant of an AD, because the pathology will be a Lewy body pathology. So that is why we term it as an atypical variant of AD. So out of which we have given here, which is an atypical variant of, which is not an atypical variant of AD. So the answer is actually semantic variant of aphasia. So I'll tell you why the answer is so. So the atypical variants of AD are actually frontal variant. That is, the Alzheimer's pathology starts in the frontal area, then slowly goes to the memory part. Then Benson syndrome is other name of the posterior cortical variant of AD. So I just put, for your knowledge, I just added it. Benson syndrome, actually posterior cortical variant of AD. It was named by Benson. It discovered by Benson. That is why it is otherwise known as Benson syndrome. And logopenic variant of aphasia. Previously, this progressive variant of aphasia was put under frontotemporal dementia. But now, because of pathology is due to kind of Alzheimer's pathology, it is now regrouped as a logopenic. This logopenic variant of aphasia is regrouped now as a Alzheimer's pathology. That is Alzheimer's variant, and not as a frontotemporal variant. And semantic variant of PPA, primary progressive aphasia, and the non-fluent variety are actually part of FTD, that is frontotemporal dementia. So in this, the answer is actually, which is not a typical variant of AD, is actually semantic variant of aphasia. So these are the typical variants. You can read through. This is actually there in Harrison itself. They are written. And posterior cortical atrophy, because the occipital, there will be occipital atrophy, and you can see all sorts of syndromes. Balance syndrome. Actually, one patient had came to me, and patient had severe visual impairment, and we had initially sent the patient to ophthalmologist thinking that patient has visual problem. It can even mimic a malingering, because a ophthalmologist will send us back the patient saying that the patient doesn't have any problem. And the frontal variant of AD will mimic a frontotemporal dementia, and like that. So that is regarding the atypical variants. So we were discussing previously regarding the genetics of AD, in which we had told that actually early onset Alzheimer's disease, the most common variety cause was actually presenilin 1. That is due to the which is present in the chromosome 1. So my question would be, actually in early onset Alzheimer's disease, when will you call it as early onset Alzheimer's disease? So any Alzheimer's disease before this age, you have to better go for a genetic testing, look for a family history. If you have time, read the book Still Alice. It has a lot of important details regarding early onset Alzheimer's disease. So whether it's 40, 45, 60 or 65, the answer is actually 65 years. Okay, the answer is actually 65 years. So this is a repeat question which is asked many times. Which is the most common psychiatric manifestation of Alzheimer's disease? So try to answer this. Is it apathy, agitation, anxiety, anhedonia? The Alzheimer's patient coming in front of your OPD, will you find him agitated, anxious, or apathetic? Usually you have to address this part of this Alzheimer's patient also, not alone treating the patient with donepezil and memantine. We should address this apathetic part also, otherwise patient will have serious problems. Also there will be problem for caregivers also. So apathy is the answer. It's actually a direct word taken from Harrison, and it's actually a repeatedly asked question. So it's about from studies which had shown that 72% had apathy. Agitation is there in 60%. So regarding the treatment aspect, this I'm dealing just with the basic things in dementia, and just with the Alzheimer's and the basic. The other advanced part we will deal in the next class. So stay tuned for the next class. So I just wanted to prime you regarding dementia and Alzheimer's disease. Which of the following is not a cholinesterase inhibitor? We had seen that because of the damage to the cholinergic neurons in the pathophysiology, there is actually decreased transmission, that is synaptic transmission, and there is actually memory impairment. So we try to give cholinesterase inhibitor so that more choline is there in the synapse, and there is increased memory, improvement in memory. Answer is actually memantine. Memantine acts through NMDA. So one more drug used to be there previously. Can you answer in the chat box which was the drug previously there, which was removed due to hepatotoxicity? Previously, while studying pharmacology, we used to remember these drugs as Dr. TG. That is donepezil, rivastigmine, T for one drug, and G for galantamine. These were the cholinesterase inhibitors. So that is tacrine. Tacrine was removed due to hepatotoxicity. And this is actually memantine, NMDA. So glutamate release, the glutamate excitotoxicity. So that is, so this table you try to remember. It's a short table. Any questions can be asked, especially in institutes. If some question regarding treatment in this is to be asked, this can come from this table. So have you heard of these two drugs, Tarenfubril, Fulbril and Semagestat? So these two drugs are actually they're in Harrison. They have given in Harrison's. There are actually two trials were going on. There was a lot of optimism regarding these two drugs in Alzheimer's disease. During the phase 2, it was thought that it will change the course of Alzheimer's disease. But later, during the phase 3, actually it failed. So because there was less of brain penetration, so that was the reason. So we have already seen where in the pathophysiology where these had acted. So these are actually acting on the gamma secretase inhibitors. So I think we have summed up everything regarding. So we have covered most of the Harrison part in detail and we have covered most of Harrison in a short period of time. In Dementia Advanced, we'll do it next level studies regarding dementia, so that we crack into the institute level of studies we can see. So thank you, everyone. I hope you had got the concept correct. I just that pathophysiology of Alzheimer's disease is very important. Please again read that part again in Harrison or Bradley, or see this video again and get a good thorough knowledge of it.